Hello and welcome to the 5th episode of the Repligen 2022 webinar series. My name is Dervla Campbell, Marketing Manager at Repligen. This webinar is one of a series that will describe a range of topics for upstream intensification and includes summaries of key historical milestones, technical considerations important for planning, guides to optimization, and case studies of example efficiency gains. Today's webinar covers increasing vaccine production by intensifying both the N and N-1 bioreactors. We're thrilled to have two industry experts join us today for the case study. Repligen's own Shelley Parra, Senior Director of Product Management, will start today's presentation to provide context for the technology and the overall study. Our guest is Hans de Hoek, Biotechnology Solutions Product and Platform Steward at MSD Health, and he'll then present the motivators and results of this team's upstream intensification work. At this point, let's cover a few housekeeping items. First of all, the webinar is being recorded so that people who can't join us today can view it at a later time. Second, we do have interactivity on the webinar platform. In the panel on the side, you'll find a single poll question. We'd appreciate it if you would take a few seconds to provide your input because it helps guide future content toward your interests. You'll also see a live Q&A panel right next to the poll. You're welcome to submit a question at any time during the webinar. Use the upvote feature for question you like so that we know how to prioritize them. And if we don't respond to your question today, you'll get a response via email within 24 hours. And with that, let's turn it over to Shelley and Hans for their presentation entitled Upstream Process Intensification for Virus Vaccine Manufacturing Case Study. Thanks again for joining us and enjoy the discussion.
Hi, and thank you for joining us today. My name is Shelley Parra. I am from Repligen, a Senior Director in Product Management in Technology Positioning. I'm here with Hans de Hoek from MSD Animal Health. He is part of the Biotechnology Solutions Product and Platform team. And we're going to be speaking about upstream process intensification for virus vaccine manufacturing, a case study that we did with the MSD Animal Health team in Boxmeer, Netherlands. So very excited to share the story with you today. Thank you. All right, if we look at a typical therapeutics production workflow for pretty much a protein or a vaccine, you have an upstream and a downstream process. And Repligen plays in both of these areas. So we are setting standards in biological drug production and really helping throughout the entire workflow. If you noticed at the bottom, the Repligen technologies in use today across that workflow. Since 2010, we have had 13 strategic acquisitions and 10 disruptive technologies that really has changed how we've been able to support customers and really help with intensifying their process with innovative solutions. Today, I'm going to talk about one particular technology and one solution, and that is our ATF. So if we look at XCell ATF, it is a cell retention device, and it's used for high density cell culture and process intensification. So high seeding for in N-1 perfusion, used in the perfusion stage, as well as other applications like high performance harvest. It can be used in batch or in continuous processing mode. It has been proven on greater than 25 different cell types, including CHO, MDCK, insect, E. coli, and stem. It has been proven scalability. It's a linear scalability from 1 liter to 5,000 liter. And we offer this in two formats, a single use and a stainless steel option. And this is what we're going to be speaking today about. And how do we go about supporting this in the field and making sure our customers are successful? And that's what I would like to speak about next. So Repligen provides a guided implementation to ensure success. So we have field application scientists that are in the field and support from consultation all the way to execution and implementation. So we go in, make sure the technology is fit. What are the solutions that you need? What are the challenges that you're experiencing? We help put together an experimental plan, help evaluate that, help optimize where needed, in the development, the sizing, the scaling, the transfer to manufacturing, training of manufacturing operators, and really help with that implementation. So throughout the entire adoption of the technology, we have scientists there to help. So today we're going to talk about one case study with MSD and how we put this this implementation process together with them to make sure that they were successful. So the case study is on upstream process intensification for vaccine manufacturing. We have a white paper on this available on our website, and this presentation here is a subset of that information. So next, I would like to pass this on to our special guest, Hans de Hoog, and he will be presenting on his work that he did at his site.
Thank you, Shelley, for the introduction. Good morning, good afternoon, everybody. I'm happy to present this case study. And before I would like to do that, I would like to take a moment to introduce the company I'm working for. So my name is Hans de Hoog. I work for MSD Animal Health with the location in Boxmeer. Our company produces medicines, vaccines, and innovative animal health solutions and services, as well as digitally connected identification systems for traceability and monitoring products. And on this picture, you see a nice salmon, trout fish, but we produce products for many other animals as well, like ruminants, poultry, swine, aquaculture, and companion animals. My company has offices in more than 50 countries and in commercial presence in more than in about 150 markets. And we are dedicated to the animal health, manufacturing and R&D sites all around the world. And my location is Boxmeer in the Netherlands. So then I move to the case study where we, in cooperation with Repligen, we tested their XL ATF perfusion technology platform. And that's the work done by my team, not by myself. So I would like to acknowledge Bas, Maarten, Teun, and Kate, who did the hard work on this project. And of course, I would like to thank Earl Pineda and Thijs Mimpe from Repligen because we work so closely together on this project. So we had main objectives. So our company would like to meet the increased vaccine market product demand. We would like to increase our productivity and increase our efficiency. And of course, we would also like to use our existing manufacturing facilities. And with process intensification, we might be able to increase our output per batch. So those are actually the broad goals we have. Why XL ATF technology? Well, we think it's important that we have an established intensification performance in different applications, and the XL ATF technology platform has this. We think that the alternating flow technology has an advantage for the cell lines that we used. We not only have nicely single cells solutions, but we also have cell cultures that tend to be some clumpy or multi-cell clumps. So that then the ATF technology has an advantage because there's less risk of clogging of your filter. Besides that, the ATF technology has the proven linear scalability, which is important for us so that when we develop something at small scale, we can also take the step to move to pilot scale and large scale manufacturing. And also an advantage is that the large scale single use filters are available to operate under closed system conditions, which is a trend in the pharmaceutical business and also in our company. So our scope of this study was 3 studies. We did an end-stage intensification, so the cell culture prior to the virus culture, in which we showed increased cell density. We did an expedited media exchange, and we tried to simplify the virus concentration because of the very high cell density. In a second experiment, we did a scale-up of the ATF system in which we moved from the small-scale ATF2 filter to the manufacturing scale ATF10 filter. And in a third experiment, we did the N-1 cells intensification and used those cells to generate a virus production in a normal batch process. And this was done on a different cell line than the cell line from the first and the second experiment. When we're going to look at the results of the first case study, the increased end-stage intensification, then we see a very good cell performance of the cell line in this case. Let me explain the graph. So on the horizontal axis, we have the days of cell culture, and on the vertical axis, we have the cell yield. And it's on the log scale. So normally you would expect an exponential line, but it becomes linear because of the log scale. But using the log scale nicely shows you how parallel these lines are of the ATF process versus the batch process, which stops at a certain density, but with ATF we are able to continue the exponential growth while maintaining high cell quality. These cells were then used in a virus culture, and we have normalized these outputs, but in blue is the reference batch process, and in orange we have the ATF perfusion process. And what we saw that when we started the virus culture at a 5 times higher cell density, then we also got a 5 times higher antigenic mass output. So that was really great to see. So the conclusions were that we were able to produce a 5-fold higher cell density, also showed us a 5 times higher viral output. We didn't have to do anything on the bioreactor settings. So the settings like temperature, pH, aeration, etc. were common as usual in the batch process. We didn't observe any membrane fouling, which is very important because we want robust operation. And there's a low shear on the cells. So the cells showed very high cell viability, even as good as the batch process. So that was also good to see that besides the shear in the filter, it did not cause any negative effects on the performance of the cells. And comparable cell viability and viral effectiveness were shown between the batch process and the perfusion process. And we were also we could also simplify the process by removing the viral concentration process because of we were able to grow at a much higher density in the same bioreactor. It was not needed anymore to do the virus concentration in a further downstream process. So that was good to see too. Then I moved to the second case study in which we tried the same process as the first study, but then we compared the scale up from the small scale ATF2 to the large scale ATF10. And here we see the results. On the left graph, you again see the reference batch process in gray, the average of the small scale runs done in a glass bioreactor. This is the average of 13 runs. And we have one large scale in a larger fermenter. And as you can see, the lines nicely show to be parallel, which means that they have the same growth kinetics. The cells have the same behavior, and in both cases, the viability was also very good. The cells from these processes were then also used in a virus culture, and the cell density in those cultures was normalized. And when we compare then in blue the small scale batches versus the large scale in orange, then we see that even if we started the large scale run in a large bioreactor at the lower cell density at the beginning of the virus culture, the output was equal to the batch process. And again, that was a very good result that showed us that it is a feasible technology and that it can be upscaled to the large scale. We had also good support from the field application support team from Earl and from Thijs. So they helped us with the calculations when we needed to set the parameters for the perfusion, like the refreshment rate, like the surface area of filter that you would need, and the volume that you could test. So that was highly appreciated, this support. So with this, we can lower the cost of goods with equal viral antigen quality, and higher volumetric output per batch translates to a more efficient use of the equipment in our manufacturing facility. Then I moved to the third case study, in which we did a study on an N-1 stage cell line. So N-1 means that we grow the cells to high cell density, and after that we use the cells in the virus culture at the normal batch process. So the perfusion was only applied to the cell culture. When we look at the results, we see in the graph that the batch process stops here at this level. And if you would like to continue it, you should inoculate it into a next vessel at low density again. But again, with the perfusion, we were able to go to much higher cell densities. We do see some drop off here towards the end, so that could be an indication that the perfusion rate could be optimized. But in general, we were very fine with this, and also the cell viability, the cell condition, was very equal between the batch process and the perfusion process. Well, cells from these cultures were then used in virus culture, and we were able to inoculate the cells at a 3 times higher cell density than we typically do for the virus process. And to check whether these cells had a good condition, we also diluted them back to the same approximately to the same level as the batch process. And we looked at the viability at harvest and the virus yield. And in the viability, we saw that all 3 processes were very comparable. So the CPE was equal in those processes. And when we look at the viral yield, we saw that the intensified process gave us a 3-fold higher virus output. So in this case, a 3 times higher cell input also gives you a 3 times higher virus output, which was very great to see. And we had an hypothesis that would work, but yeah, we were very happy to see that this also was the case. So as a conclusion, we can say that the production bioreactor with cells from the N-1 bioreactor resulted in a 3-fold increase in cell density and also in a 3-fold increase in viral output. The intensified N-1 cells performed comparable in virus production and was also comparable to historical batch data, and that's then the gray curves. So we have fully confidence that that these cells behave exactly the same as in a normal batch process. And that brings me to the final conclusions of our of the joint case. is cooperation between our company and Repligen. So XCell ATF enables significant productivity gains in redesigned commercial animal health vaccine processes. We showed flexibility to implement a perfusion at either the N-1 stage, so only the cells pre-culture, or even at the N stage production, which means the cells and the virus culture. In our studies, we showed a 3 to 5-fold increase in cell density, and we also showed that 3 to 5 times higher viral output was feasible. This gives us an improvement in plant capacity utilization because we can use a higher cell output per bioreactor volume. Mitigation of the facility expansion needs an efficient use of process equipment. So, which means that we might use the implementation of the perfusion, and with that, we could do without further expansion of manufacturing facilities, which is very cost intensive. So we think that the ATF technology gives us a reduced cost of goods and also a lowered capex. And the single-use options provide ease of use and possibility for closed system operation, which is very important in the current pharmaceutical market and also for our company. And with that, I would like to thank the Repligen team to give me this opportunity to present these results. And I wish you all a very good day. Thank you for your attention.
Hi, everyone, and thank you for joining us for the Q&A portion of today's webinar. Both Shelley and Hans, our webinar speakers, are here with me today, and we're excited to continue this conversation with your great questions. Our first is from Mike T, and he asks about VVD values. Were they the same at both scales?
Yes, Devilla, I can take that question. Good morning, good afternoon, everybody. Welcome in this webinar. The question on VVD, yes, both at the small and the larger upscale, we used the same VVD values. That's true.
Excellent.
Yeah, and just for those that may not know what VVD is, we'll just, so that's
sorry.
Vessel volumes per day, and that's the perfusion rate that is going on in the system. Right.
Yes, and for reason that for upscaling, we try to keep everything as close as possible between the small scale and the large scale.
Great. Our next question is from Miguel, and he asks, was the MOI the same for the standard and intensified virus production processes?
Yes, I can take that question as well. So Miguel, in the experiments that we performed, we did use the same MOI in the standard process versus the intensified process. So basically we took the process parameters from the standard process and translated it to the intensified process. So that is confirmed. Yeah.
Great. Our next question, was the material taken all the way through DSP? Were any DSP changes required?
Well, in some cases we did do the complete DSP. I cannot go too much into the detail, but it is, I can say that, of course, because of the higher biomass, you have to be aware that some adjustment might be needed in the downstream process. For example, if you would like to do a clarification process, then you would need a different setup for the centrifuge, or you would need a higher filter surface for filter clarification. Yep. I hope that answers the question.
Okay. Can you confirm that you are using transient transfection?
I don't understand that question, so I don't think I can give you a good answer to that one.
Okay, no problem. Moving on.
Sorry about that. Sorry.
That's okay. Is it possible or worthwhile to intensify N-1 and N for the same process?
I think that you could do that. If it's worthwhile, will depend on the process that you're running. So I think it will be a business decision if you're going to do the N-1 and the N process. So it will highly depend on what type of cell, what type of virus, or what type of other molecule that you're running. So that's a difficult one to answer in this case.
Okay.
Yeah, if I can just expand on that as well for a second. So I think it completely is a business case, and what they're trying to intensify and where the biggest challenges are for the manufacturing. So wherever they can get their biggest bang for their buck, or the most I guess where it's the easiest sometimes to come in, N-1 is a very common application to intensify. But we are also seeing an increase for the end stage as well. So you can do N-1 and alone, or we are seeing a lot of both of them at the same time. And it really, again, just depends on the challenges and what the goals are of the company.
Very good. The next question is from Friedrich. He asks, which host cell type did you apply, and what were the cell densities pre and post intensification?
Yeah, so I cannot go into detail about the cell lines that we used in these case studies. So sorry about that. That's confidential information. I can say that what we explored was the area where we had a normal batch process, which had a certain cell density, and in this case we moved it up to a 5-fold cell density when moving into the virus infection.
And maybe I can expand on that, not from Hans' experience, but from in general. And with ATF, we have done similar intensification with over 25 different host cells, including CHO, PERSI-6, HEK293. So we have experience with lots of cell lines. And we see very similar intensification and improvements in the VCD.
Very good. Our next question, can you estimate the overall COGS reduction percentage?
I wish I could. We are still working on it. I think it could lead to a 25% cost of goods reduction. It is difficult to predict because you need to run a lot of experiments before you exactly know what the output is. You have, of course, reduction of cost because you don't need that many bioreactors. But on the other hand, you have to invest in the technology and in the also in the consumables from the ATF. So that comes on top of it. The most you will save on the man hours because you're doing more output for one batch, and that will be the biggest saving. Maybe Shelley, you can add something from your perspective.
Yeah, I think that answered the question really well. When it comes to the COGS and the improvement, we definitely see improvements, but I couldn't say like the percent as well, because that's so customer dependent, really. Yeah.
Okay.
Yeah.
Okay. Moving on. What size bioreactors were used for the ATF2 and 10?
So for the ATF2, we use a small scale bioreactor, and for the other one, a large scale. That's obvious. I cannot go too much into detail about the volumes, but we were well within the range that is recommended by by Repligen, which is the 2 to 10 liter volume for the ATF2 filter, and the 200 to 500 to 1000 range for the ATF10 filter.
Yeah, so I'll just expand. So on that, so ATF, we do have, if someone wants to go smaller, we do have an ATF1 that you can go down to 0.5 liter sizes, and we do have customers using this all the way to 5,000 liters as well. But I don't think Hans was at the 5,000 liter in this case.
Okay, Nick is asking if you're not using transient plasmid transfection, how do you initiate viral vector production? Is it inducible cells?
I'm not sure if I understand this question correctly, but in our case study, we use a classical approach where we infected the cell line with a virus. So we did not use a vector. It's the classical process of a virus on a cell line.
Hans, there it's a producer cell line, correct?
Sorry?
Was it a producer cell line, correct?
Yes.
Yes. So I don't think that you did transient transfection in this particular case. We do have lots of customers that do that, but in this example, that was not the case.
Yeah. Okay. Thank you.
Okay, and I think we have time for just one more question today, and that will be, what were both the bioreactor and filter single use?
Yeah, we tested several options. We tested the it's not multi-use, but we tested the stainless steel ATF filters, and we also tested single-use versions, and they both work very fine. And we also tested multi-use bioreactor and the single-use bioreactor, and in both cases we got good results from that as well. And
Excellent.
Sorry, I cannot go into detail about the exact volumes or the type of bioreactor that we used. Sorry. Yeah.
Great, thank you, Hans. At this point, we need to wrap up today's discussion, but we've gotten so many great questions, and rest assured, Shelley and Hans will respond to you via email as soon as possible. And next, as we mentioned before, this is the first in a series of several webinars on process intensification. And our next one is in about 3 weeks. And we'll be sharing some marketing communications on that within the next week or so, providing more information, and hopefully you'll join us again. All of you in the audience for spending some time with us today. We appreciate your time.